This is part one of the chapter 3 lecture notes. Let me set it up. All right, chapter 3 is another chapter. Well, it's our last chapter on chemistry, on basic chemistry, but it's going to focus on the macromolecules, like we mentioned in chapter 2. Chapter 2 was more basic chemistry background information that you need, and then chapter 3 will focus on the molecules of living things. Um, Sometimes it's called biochemistry, but we're going to focus on the 4 major classes of macromolecules. And this kind of overlaps a little bit with nutrition. You'll recognize the macros, carbohydrates, lipids, which are your fats and oils, proteins, and then nucleic acids, which is your genetic material. All of these macromolecules and some smaller molecules are organic. And if they're organic, the definition technically is that they contain carbon and hydrogen. They must contain carbon and hydrogen, both, or they are not organic chemicals. I know it says down at the bottom may also contain hydrogen, oxygen, nitrogen, and some other minor elements, but organic molecules always contain hydrogen and carbon. Always, always contain hydrogen and carbon. This is one thing that I found that the slide presentation and the textbook maybe weren't as clear about. But this is how you distinguish between organic molecules like the 4 types that are listed above, and something like maybe carbon dioxide, which is not an organic molecule. Carbon dioxide would be inorganic. Yes, it contains carbon, but it does not contain hydrogen. And yes, carbon dioxide is found in living things. In fact, it's a byproduct of cellular respiration. Um, and it's the gas that we breathe out whenever we are breathing. We breathe in oxygen and we breathe out carbon dioxide. Carbon dioxide builds up in our tissues as we break down glucose for energy, but it's not an organic molecule, it's inorganic. So just to make that clear, if it's organic, it's going to have carbon and hydrogen. Um, the 4 classes of macromolecules, we're going to kind of have a pattern. Each macromolecule will consist, it'll be a chain of individual links in the chain or individual building blocks, which we call monomers. So the smaller subunits are monomers, and then the monomers linked together by covalent bonds are called polymers. So for example, the building blocks of proteins are amino acids. So an example of a monomer would be amino acids. And then the protein would be the polymer. So monomers are the small subunits, the building blocks, and then polymers are the chains that are produced from the monomers. So in this example, you see a disaccharide being formed. Each one of these hexagon shapes are monomers. They are simple sugars or monosaccharides, and these two happen to be the exact same monosaccharide. They're 2 molecules of glucose that are being joined together to form maltose. Maltose is a disaccharide. Di means 2. So you've got 2 monosaccharides called glucose that are being joined by a covalent bond or a glycosidic bond to form a disaccharide called maltose. And so that shows you kind of how monomers are linked together to form polymers. You also can see that a water molecule is removed in the process. Hydrogen is removed from one of the glucose molecules, and OH, or hydroxide, is removed from the other, and together they form water. So that's why we call this a dehydration, that's removal of water, synthesis, which means to build. And then the opposite of that would be hydrolysis, which is breakdown. Lysis means breakdown. So it is the breakdown of a larger molecule, or the breakdown of a polymer into individual monomers. So this is a mistake. I'm so sorry. I should have proofread these slides. It's a breakdown reaction. So dehydration synthesis is a dehydration reaction, which means removal of water. Hydrolysis is a breakdown reaction. I guess that's what you can just write that in place of dehydration. I apologize for that. So in this case, maltose, which is a disaccharide, is broken apart with the addition of water. This is the opposite of what we saw happen in the dehydration synthesis. An OH is added to one of the subunits to form glucose, and then an H is added to the other subunit to form another molecule of glucose. So maltose is broken down into two glucose monomers. And also, if you would like to watch this little video clip, that is slide number 5. We're going to move on to slide 6. But the video clip shows chemical reactions involving monomers and polymers. All right. We also have to talk about enzymes in this chapter, because enzymes are necessary for every chemical reaction in the body. So an enzyme is technically a protein. It's a macromolecule. It's a protein. Enzymes are never any other type of chemical. They're always proteins. And what they do is they speed up chemical reactions, and we call them catalysts, because catalyze means to speed up chemical reactions. So enzymes will speed up hydrolysis and dehydration reactions. Dehydration reactions form new bonds, and they require energy. We call them endothermic. And hydrolysis reactions break bonds, so they release energy. This is what we do when our cells break down the bonds in a molecule of glucose in order to get energy out of the glucose. That is called cellular respiration. But hydrolysis reactions occur when polymers break down into monomers. And every time a hydrolysis reaction occurs, energy is released. There are specific enzymes for each macromolecule. So carbohydrates can be broken down by several enzymes. This is a very short list of enzymes. Amylase breaks down the carbohydrate amylose, which is a starch. Sucrase breaks down sucrose, which is a disaccharide. Sucrose, you may recognize that. That is the name for table sugar. Lactase breaks down lactose, which is the sugar found in milk. And maltase breaks down maltose, which is the sugar found in malt beverages, malt liquor, and malt that's found if you order a malt, like a chocolate malt instead of a chocolate shake, the malt powder that they put in there, that's maltase sugar. Okay, lipids are broken down by lipases. Proteins can be broken down by pepsin and peptidase, and also various forms of an enzyme called trypsin. Okay, so we're going to start with carbohydrates and talk about carbohydrates, different examples. You probably know examples of carbohydrates from nutrition. You know that they can be breads and pastas and grains like corn and rice, quinoa. So these are all examples of carbohydrates, but you also have carbohydrates in fruits and vegetables. And of course, different types of sugars are carbohydrates. So carbohydrates are found in grains, fruits, and vegetables. Carbohydrates provide energy to the body in the form of glucose. So even if it's a carbohydrate like starch, it will break down into glucose, and your cells will use that glucose for energy. Carbohydrates have a specific ratio of carbon to hydrogen to oxygen. It's always 1 to 2 to 1. So for example, the formula for glucose is C6H12O6. That's 6 to 12 to 6 or 1 to 2 to 1. So for every carbon and for every oxygen, there are 2 hydrogen atoms. The subgroups of carbohydrates are monosaccharides. Those are the single simple sugars like glucose. Disaccharides, which are a combination of 2 monosaccharides. And then polysaccharides like starch, which are many sugars, many monosaccharides linked together in a chain. Some examples of monosaccharides are aldoses and ketoses, but that may not mean anything to you. So let me give you some names. Glyceraldehyde, ribose, glucose, probably the only one you've really more than likely heard of is glucose. If the sugar is an aldose, then it has a C double bonded to an O. That is called a carbonyl group. So when the C double bonded to the O is on the end, it's called a carbonyl group. So aldoses will have that. Glyceraldehyde is an aldose. It's also a triose because it contains 3 carbons. 1, 2, 3. Ribose contains 5 carbons, so it's a pentose. Ribose is another aldose because it contains that carbonyl group. So these 2, glyceraldehyde and ribose, are aldoses. And glucose is another aldose because it contains a carbonyl group. So let's look at one that would be a ketose, which would contain the carbonyl group in the middle. And fructose would be an example of a ketose. So here you can see 2 aldoses because they contain the carbonyl group on the end. And then you can see a ketose sugar called fructose because it contains the carbonyl group in the middle. All of these, glucose, galactose, and fructose, are examples of monosaccharides, and they contain 6 carbons, so they are hexoses. Many times they form a hexagon shape. Glucose is blood sugar. Galactose is a monosaccharide that helps to form lactose found in milk. And fructose is a sugar found in sucrose in fruit.